Before we get started, we'd like to especially thank Thermo Fisher for sponsoring this session. Thermo Fisher Scientific is the world leader in serving science. Their mission is to enable their customers to make the world healthier, cleaner, and safer.
Hello, good evening, or good evening from here in Vietnam from me. I'm Louise Thwaites, and I am moderating this session 13 of the World Sepsis Congress. I think it's a really exciting session. We're looking at novel approaches in the diagnosis of sepsis and COVID-19. But before I start, I would like to thank the rest of the organizing committee for bringing this all together, and to Thermo Fisher, who are our exclusive sponsor for this session. So just a tiny bit about me. I'm a clinician scientist based at the Oxford University Clinical Research Unit here in Ho Chi Minh City, Vietnam, with a special interest in critical care in low resource settings. I am in the steering committee for the Asia Pacific Sepsis Alliance, and I lead their guidelines and policy group, and also a member of the Asia Critical Care Network, ICU network. If you've been with us for the last 2 days, I think you will have heard many of the speakers talking about how timely diagnosis and accurate diagnosis of sepsis is really important to us achieving our ultimate gain of improving sepsis outcomes. So I think today's and tonight's session is particularly important because of that. We've got a really great lineup of 6 speakers. They're all kind of leading very diverse fields. We're going to move from AI to genomics to implementation projects. And I'd really encourage you to use the chat box. Please put questions forward. They're a really nice group of speakers, and make the most of this opportunity to interact with them. So the format of the session, we'll have about 10 minutes or so for each talk, and then a few minutes for question and answers. So again, I encourage you to interact, put your questions in the box. So with that, I'd like to move on to our very first speaker, and that's Professor Philipp Schütz. Philipp is joining us from Switzerland. He is an internist and an endocrinologist with a special interest in clinical nutrition. He's got very broad clinical and research interests from the endocrinology, infectious diseases, critical illness, and general medicine. And he's done a lot of research in the field of nutrition, hormones, and biomarkers. He has a professorship from the Swiss National Foundation, and he currently works as head of the Emergency and Medical Department at Medical University Department in the Kantonsspital Aarau in Switzerland. So I welcome and introduce Philipp Schütz.
Well, thank you, Louise, and welcome from my side as well. It's great to be here and to have the first speech today about the evidence for the role of biomarkers for antibiotic stewardship. Here you can see my potential conflict of interests. And I think if we talk about antibiotic stewardship, there is a lot of different key elements to that. It's about supporting an interdisciplinary approach. It's about education. It's about controlling source of infection and other things. But I think where really biomarkers can have the most impact for antibiotic stewardship is for 2 key elements. And this is first, prescribing antibiotics when they are truly needed. And second, using the shortest duration of antibiotics based on evidence. I think here really biomarkers can make a difference. So if we talk about biomarkers in general for sepsis, I think we talk about 2 different types of markers. So one of the markers are host-directed biomarkers, which would be these levels such as C-reactive protein, which is an inflammatory marker, or procalcitonin, which is more an infection marker. And they tell you information about how the host is responding to infection. The other types of marker are then more the marker of the pathogen itself. So we can have cultures, we can have PCR tests. And I think the big challenge for us, if we want to manage a patient with sepsis, is how can we add all this information in a meaningful way to actually have a benefit for our patient. So I think most importantly, what we have to do is we have to think about the patient. We have to integrate all the information we have into the big puzzle of the patient. So always first, we have to do a pre-test probability for the patient. Is it a bacterial infection or is it rather not a bacterial infection? Most of the patients we see in the emergency department or ICU on the first day of admission, we are not sure about it. But we have to make an assessment. Are we certain or uncertain? Because based on the pre-test probability, our biomarker is going to help us differently. So let's say we have a patient and we have uncertain bacterial infection. We don't think it's bacterial infection, but he looks inflamed. And then we have the low biomarker. Then it would tell us, well, bacterial infection becomes extremely unlikely. Now, on the other hand, if we see a patient where sepsis is highly likely, then we have to be more prudent and we have to look at the biomarker differently, because as we all know, biomarkers are not 100% accurate, and though we have to add it to our clinical and microbiological workup, and then it can also influence antibiotic management. So only in a patient where we have a low pre-test probability for bacterial infection and a low biomarker level, we may not want to use antibiotics on admission. And in a patient where we have a high clinical likelihood, we may still overrule the biomarker just to be on the safe side and may use the biomarker later on to de-escalate treatment. So if we use the biomarkers such as PCT, then what is the clinical benefit for the patient? And there has been a lot of different trials that have compared biomarker-guided care to patients with routine care. And we have done some work, some meta-analytical work together with the Cochrane Library. We have gathered a lot of these trials and we were able to obtain individual patient data. So of all these patients included in these previous trials. And we have done a couple of different analyses to see, well, what's the effect in regard to consumption of antibiotics, in regard to clinical outcomes. So taken together, when you look at all the patients today that have been included in such trials for respiratory tract infections, you see this is what you get in regard to antibiotic consumption. And you see we have two different effects. We have lower use of antibiotics on admission. And this is particularly true if you have patients with a bronchitis type syndrome, so COPD exacerbation, bronchitis. These patients typically have lower antibiotic use with the use of a biomarker approach. But also you have for the typical sepsis patient, which you are going to treat on admission. For this patient, you can have a shorter duration of treatment and thereby reduce overall exposure. And you see in this analysis, which was focused on respiratory infection, we had overall a reduction of mean antibiotic usage from about 8 to 5.7 days. So it is quite a reduction in antibiotics. Now, more importantly, maybe for us than antibiotics is also the question, well, how about the clinical endpoints? If we shorten antibiotics, do we hurt the patient in the end? And I think there have been some very important trials looking at this. And the largest trial today comes from the Netherlands, from Evelien de Jong and her group, the SAP study. And you see, this was a large study with over 1,500 patients with septic infection in the ICU. And the author found not only a reduction in antibiotic use, but actually there was a reduction in mortality. So in this trial, if you had standard care, your risk of dying was about 25% increased compared of the biomarker approach. We had similar results also in the meta-analysis, so taking all these trials together, where we had about a 20% reduction in the risk of mortality, of treatment failure, and also we had a lower risk of antibiotic side effects, which of course was associated with the lower antibiotic use in these patients. So how about PCT and COVID? And I think this is an important question because COVID, of course, is a very severe lung infection. And for some patients, we are not sure whether it's only COVID infection or whether it could be super infection, particularly for patients remaining in the ICU for longer time. Maybe they have another spike of CRP, they may have another fever episode, and we're asking, well, could it be infection? So I think PCT has been studied here in a number of trials and studies. And I think so far we have two main conclusions. So the first one is that PCT in the context of a COVID infection provides prognostic information. And so here you see that there have been a couple of different risk scores. There is one which I think is pretty convincing, which was just published in the Clinical Infectious Disease end of last year. And so the author found that actually 5 different markers, including neutrophils, lymphocyte, PCT, CRP, and the age of patients, all together gave a pretty high prognostic accuracy. And you can see that a patient with a low risk based on this score had an almost 0% mortality rate, while if you had an increase in the score, your risk would go up to about 40%. So prognostically, I think an increase in PCT gives you prognostic information. But there is also some studies looking at PCT as an antibiotic stewardship guide. I have not seen randomized studies, but I have seen studies looking at patients where PCT was used during routine care. And I think this is one of these studies I would like to share with you. And you see the authors used a similar approach I have just shown you. So looking at the patient, is it clearly sepsis? They would give antibiotics without considering any marker because it is a dangerous disease. But if not, they would hold off. And then they would use PCT as well as the clinical course to decide to early stop antibiotics. And also in this study, they used the PCT cutoff of 0.25. So a patient clinically responding, having a level below that PCT of 0.25, they would advise against use of antibiotics and for the stop of antibiotics. And so basically the results here, they suggest that in patients where PCT was low or dropped, there was a much lower use of antibiotics compared to these patients with higher PCT levels. So the study concluded that the low PCT was helpful in the de-escalation of antibiotics in the COVID-infected patients with possible superinfection. And I think that's one of the big rationales that people have been using. So taken together, what's the evidence for PCT in sepsis and in COVID? I think for sepsis, we do have strong data, particularly for the respiratory tract infections, pneumonia, bronchitis type infections, COPD exacerbation, but also for surgical patients and for patients coming in with different sort of sepsis. I think here we do have the strong randomized trials. Now for corona, for COVID infections, I think there is some interesting studies out there, but we do not have the large randomized evidence today that we want to have before widespread implementation. But what I have seen so far regarding prognosis and also the stewardship studies have been promising, and I'm sure we will learn more in the next weeks and months how to best use the biomarker approach to manage patients with COVID-19. And with that, I'd like to close and thank you again for your attention.
Okay, thank you very much, Philipp. That was really excellent start to our session, and followed on really nicely from the previous session looking at antimicrobial resistance and sepsis diagnosis. I'm just looking at the chat here. There's a quick question. I think this might be quite hard for you to answer, but I'm going to ask it anyway. What do you think is the PCT cutoff value for an antibiotic?
So actually, the cutoffs that have been used mostly is 0.25 for the patient with a typical respiratory type infection. And for the very severe septic patient, the cutoff that was used mostly is 0.5. So let's say, in my experience, for the COVID patients, I have seen most of these patients actually coming in the hospital with a very low PCT level, so 0.1 or even lower than that. If during the course of disease the patient, deteriorates and his PCT spikes up, and I have seen patients with superinfection, and they will go up higher than 0.5 or higher than 1 or 2 or even higher than that. But in the normal COVID patient that is going smoothly without problems, PCT remains quite low in my clinical experience so far.
Okay, thanks very much. I'd like to follow on. I work here in Vietnam, and as you probably know, PCT is quite an expensive investigation in a lot of countries. Do you have any idea whether there have been some cost-benefit studies done or anything that might suggest it is worth implementing in lower resource settings, particularly if it saves antibiotic use?
Yeah, that's a great question. Actually, there have been a number of cost-benefit studies, and I think the studies show if you have a patient, let's say in the ICU, and you spend and you can reduce your antibiotics by 2 or 3 days, and antibiotics are very expensive, and if you can reduce that by 2 or 3 measurements of PCT, then this is clearly a cost-effective strategy. Because you have lower antibiotics, you have also some benefits in regards to complications, maybe C. diff and other things. Now, I think what is important is that we use PCT only in situation where actually the biomarker can have an impact on the clinical decision making. So if we do know that we will use antibiotics anyway, or if we do know we are not going to use antibiotics anyway, I don't think the biomarker is going to add a lot. And in this situation, it's not going to be cost effective. So we should focus really on these patients, these situations where we believe the marker can add, to our clinical management. And there I see respiratory tract infections, and also ICU patients where we can shorten the duration. I see these as the major population where we will see cost benefit.
Okay, a couple of quick questions in the chat there. Firstly, do cutoff vary in patients with cancer?
No, actually not. I mean, there is some types of cancers such as C-cell carcinoma of the thyroid, where the cancer is just producing procalcitonin, but this is quite a rare situation. But normally, in a patient with cancer, you would expect that this patient has low PCT, unmeasurable PCT, and only when he's infected, his PCT increases. But there you can use the normal cutoffs. Okay,
thank you very much, Philipp. I think actually we're going to have to move on now. I think you can interact directly. There's still one more question in the chat there. So thank you very much, Philipp. I'm now going to introduce our next speaker, and Philipp's talk really has brought up this issue of biomarkers, and I think this is now really going to be developed through the next couple of talks as we move into the field of precision medicine. And our first speaker on this is going to be Professor Tom van der Pol. He is a specialist in internal medicine and infectious diseases. He's a professor of medicine and chair of the Department of Medicine at the Amsterdam University Medical Center. He's former chair of the International Sepsis Forum and was actually a member of the international committees that established the new sepsis definitions in 2016 and the surviving sepsis guidelines. His research focuses on pneumonia and sepsis, and particularly the areas of host response and pathogenesis. And today, Tom is going to talk to us on how precision medicine is going to change sepsis management. So I welcome Tom now.
Thank you very much, Louise. So this is my first slide about how precision medicine will change sepsis management. What this slide depicts, and I'm not sure whether the animations are visible, but if not, then I'll guide you through it. In the upper part of this graph is the host response depicted as primarily pro-inflammatory, whereas in the lower part in blue, that's the anti-inflammatory part of the host response. And what I depicted here in the upper part is the fact that for decades we've been doing trials seeking to inhibit the pro-inflammatory response of sepsis, and all of these have been more or less off target. The primary endpoint of all of these trials was mortality, usually 28-day mortality, and all of these trials failed to accomplish a benefit with regards to this primary endpoint. Now, what you have to realize that all of these trials used what we call now prognostic enrichment, and which means that the patient population that was enrolled in these trials were enriched for the primary outcome. So they were basically very severely ill, the high likelihood of dying, because otherwise you have to design trials with thousands and thousands of patients, because you will never be able to show benefit if the primary endpoint is not that prevalent. So patients were not selected based on the likelihood of which they would benefit from a certain intervention. They were basically selected on severity of disease. And this is depicted on this slide also, which was actually made by the next speaker, Hector Wong. Here on the left part, you would see prognostic enrichment. So these patients that are heterogeneous, the sepsis populations are stratified according to their mortality risk, in a low mortality risk and a high mortality risk, and this is what we call prognostic enrichment. Now, the patient with low mortality risk in the upper part could be treated with standard care. However, the patients with high mortality risk could be further stratified into subpopulations that are more likely to benefit from a certain immunological or biological intervention. And this is what we call predictive enrichment. So enrichment of the population with patients that are more likely to benefit from a specific intervention. This is an example of a recent sepsis trial that sought to use predictive enrichment. And this is the trial looking at soluble thrombomodulin in patients with sepsis-associated coagulopathy. So what I show here is that patients were selected based on sepsis, but also based on some laboratory values that would indicate coagulopathy. And this was depicted here. Unfortunately, even though predictive enrichment was used in this trial, there was no signal. As you can see, the far right end of this slide, there was no benefit with regards to survival. Now, if you go back again to this pathophysiology slide and looking then at the lower part of the slide, and it's a little bit obscured because the animation is not there, but I highlighted there some of the key features of anti-inflammation in sepsis, which in the end can result in immune suppression. And there has been many investigators out there that now advocate the use of immune-enhancing therapy rather than anti-inflammatory therapies, such has been evaluated in the past decades in clinical sepsis trials. Now, if we look at potential interventions that might enhance immunity in patients with sepsis, so seeking to reverse the immune suppression that sepsis patients usually suffer from, the intervention listed here, interferon gamma in the upper left corner, GM-CSF, and also interventions targeting programmed cell death, one or the ligand thereof. IL-7 has also been evaluated in patients with sepsis, and IL-7 mainly targets lymphocytes. Now, some of these trials have indeed used predictive enrichment, and this is an example of a relatively old trial that evaluated GM-CSF in patients with sepsis, and patients were only enrolled if they had signs of immune suppression as reflected by a low monocyte HLA-DR expression. That's the red arrow on the top part of the slide. So only if they had this low expression, which is indicative of immune suppression, they were enrolled, and patients with normal HLA-DR expression on monocytes were not found eligible for this trial. It was a fairly small trial, as you can see, only 38 patients. And what's also nice and illustrative of precision medicine, then you give the intervention, and this readout, the readout based on which you selected the patients for this treatment, can also be used for treatment monitoring. So you have to realize that many of the anti-inflammatory strategies that were tested in patients with sepsis did not have such a readout. So did not have a readout that monitored the immunological effect of the intervention. Now, in this case, they monitored on the left side HLA-DR expression, and sure enough, patients treated with GM-CSF in the black dots showed an increase in HLA-DR expression, whereas those not treated with GM-CSF did not show this. And also another sign of immune suppression was reversed. That's the reduced capacity to produce tumor necrosis factor, by blood leukocytes upon stimulation with lipopolysaccharide, gram-negative bacterial component. Clinical endpoints, the trial was not powered for this, but there was some non-significant benefit with regards to mortality. This is another example of predictive enrichment, interleukin-7. Only patients enrolled in this trial with lymphocytopenia and not patients without that. So lymphocytopenia is also considered to be a marker of immune suppression, and IL-7 specifically targets the lymphocytopenia. That is, it's associated with an increase in lymphocyte counts, as you might appreciate from the lower graphs that shows the effect of IL-7 in the colored lines, so the red and the blue lines. Okay, now more recently there have been an interesting set of studies focusing on the so-called macrophage activation-like syndrome, which is a marker of a reflection of hyperinflammation in sepsis and also in other diseases. And the presence of macrophage activation-like syndrome might help in selecting those patients that might benefit from anti-inflammatory therapy. And the excitement primarily came from this retrospective analysis of a trial with interleukin-1 receptor antagonist, so anakinra, an intervention inhibiting IL-1 effects, so an anti-inflammatory strategy. The trial was published in the 90s, totally negative. But when they did a retrospective analysis of this trial, they found that patients that had evidence, laboratory evidence of a so-called macrophage activation-like syndrome did benefit from interleukin-1 receptor antagonist treatment. And this is in the lower bullet point. So the presence of this macrophage activation-like syndrome is based on hepatobiliary dysfunction and disseminated intravascular coagulation. And if we only look at these patients, they benefited from IL-1RA. So this has raised enthusiasm that predictive enrichment might also work in patients with sepsis. And I'm going to provide the example that of a trial that has just been completed. Results are not available yet, where they stratified patients based on a pro-inflammatory phenotype or an immune suppressive phenotype. So pro-inflammatory phenotype was defined as a ferritin level above 4420 nanograms per mil. And in a separate paper, the Greek sepsis group showed that this was a reliable marker, sensitive and rather specific for macrophage activation-like syndrome in patients with sepsis. But patients that had a low monocyte HLA-DR expression were stratified to an immune stimulatory therapy. And which therapies did they choose? These are the PIs, Evangelos Gemeros Bourbulis and Mihai Netea. They treated with interleukin-1 receptor antagonist if the ferritin level was above 4,000. And they treated with interferon gamma if the patient had signs of immune paralysis, that is HLA-DR expression that is low. So this is a classic example of predictive enrichment. They now treat the patients not as a whole group, but they select the patients, stratify the patients, and then base the specific immunological intervention on the likelihood by which they might benefit from this intervention. So this is already getting to a summary slide. What I've tried to show you is that early trials almost exclusively used only prognostic enrichment, so they only selected severely ill patients with a high likelihood of dying and did not take into account biological mechanisms that were dominant in this patient population. Now, what precision medicine or personalized medicine implicates is that we do predictive enrichment of the heterogeneous sepsis population, where we stratify patients based on certain biomarkers or profiles in subgroups that are more likely to benefit from a certain intervention. I've given you the example of ferritin and monocyte HLA-DR expression as biomarkers that might help to stratify patients, and also lymphocyte exhaustion markers can be used to identify patients with immune suppression. And more modern, and Hector Wong is one of the main investigators that have done this, you can also do unbiased approaches, and thus far, in particular, transcriptomics of blood leukocytes has been used in this area, but you might also envision that other omics fields like lipidomics or proteomics can be used, metabolomics, to identify patients in a more unbiased way that belong to a certain group biologically, within this entire big group of sepsis that is very heterogeneous. So in summary, what I've tried to convey the message that the one target fits all principle should be abandoned in sepsis. This is a very heterogeneous syndrome, and it's unlikely that all patients would benefit from a single immunological intervention. These patients can be stratified into subgroups that are more likely to benefit from a certain immunological intervention. And obviously, we need the answers quickly, so we need bedside tests with computational support and possibly supported by artificial intelligence to stratify those patients in more homogeneous groups. And this might really advance the field and make precision medicine possible, even in the field of sepsis. Thank you very much for your attention.
Thank you very much, Tom. That was a really comprehensive and very clear talk. And I think you've introduced very nicely this concept of the problems we have in sepsis management and diagnosis because of the heterogeneity of the population. And I know we're going to be coming back to it later in this session as well with some of the other talks. A question actually in the chat, which probably we can use for many speakers, was are there any biomarkers we can use, or I guess we could extend that to other markers that can give a fast response? I think probably the question was designed for immediate use in the clinical setting.
Well, the very short answer is no. So the previous speaker spoke about procalcitonin, which has been used basically mainly to stratify patients to bacterial infection or non-bacterial infection. But that's not really the topic of this talk or precision medicine. Molecular markers that have been evaluated in literature, and that's RNA profiles, can be transferred into bedside testing because PCR-based testing tests are already available. Only the contents needs to be decided upon. So there's a couple of molecular signatures out there based on transcriptomics data that have not been prospectively evaluated thus far, but hold promise for future use maybe in the clinics. So one of these sepsis signature identifies patients that are harmed by corticosteroids, for example. And Hector Wong has looked at pediatric sepsis, also looking at signatures that might tell you which children might benefit from corticosteroids and which children do not benefit from this treatment. There's also evidence from the ARDS literature. Cameron Calvin has done a lot of work in this area, and she also developed signatures based on proteins, mainly protein biomarkers, to identify patients that might benefit, for example, from statin therapy, or from or can be harmed by certain fluid challenges. So in clinical practice, not in use yet, but I think we will see that in the future.
Great, thank you. That's really helpful. So we're now going to move on to our next talk. So I'd like to say again, thank you very much for Tom for that talk. We're moving on. We're going to stick with the theme really of heterogeneity, and we're going to move on to Purvesh Khatri, who is our next speaker and is an associate professor at the Institute of Immunity, Transplantation and Infection, and the Division of Biomedical Informatics Research in the Departments of Medicine and Biomedical Data Science at Stanford University. His research focuses on developing machine learning methods to interrogate large and very heterogeneous data sets, but real-life data across a broad spectrum of diseases, and particularly aims to define disease signatures which are relevant for diagnosis, prognosis, and mechanistic understanding. So Purvesh is going to be talking particularly around the mRNA signatures in sepsis, and hopefully giving us some insights into this and the disease itself.
Thank you. Thank you for inviting me, and thank you for the kind introduction. So I was given a topic to talk about mRNA signatures that have been observed in sepsis and how it allows us to look into causative organisms. So this audience doesn't need any introduction on how big the problem sepsis is, or that majority of the patients, we cannot detect the causative organism despite significant advances in different technologies for detecting a pathogen. But there are a few advances recently that really allow us to start thinking about pathogen identification in different ways. So number one, this is not recent. This is the advance that's been going on for a few million years, and that's the immune system. If you think about what an immune system has evolved to be, it's a distributed sensor with a built-in amplifier. And what do I mean by that is that this is a host response that continuously surveils every organ and has been continuously evolving to precisely identify different pathogens and how to respond to them, even when they are not in the bloodstream and they are isolated in a specific organ. So one thing to think about that we can start taking an advantage of is asking immune system, hey, what are you looking at? And use that to improve our diagnosis and prognosis of a patient with an infectious disease. The second advance, and that's relatively recent in the last 20 years, is starting to think about how to leverage the heterogeneity that exists across different data sets. So if you think about traditional biomedical research paradigm, we usually start our research with a relatively homogeneous group of patients. So for example, blue people. And the traditional approach has been to discover a biomarker in this relatively homogeneous patient population, show that in the same patient population, it continues to perform. And then before we can go in a clinical trial or a clinical practice, show that it works in other patient population and it starts to fail. So the results, because this entire approach of using a single cohort doesn't represent the real world patient population heterogeneity and lacks the generalizability. But since the microarrays came onto the scene, what's happened is these data are continuously collected in public domain. So now what is possible is to take multiple data sets that come from different cohorts from different countries generated by different investigators in different patient populations and integrate them into single analysis. And what this allows us to do is it allows us to account for biological, clinical, technical heterogeneity between and across data sets, identify more generalizable solutions that increase the a priori probability of continue to perform just the way they are intended in other unseen patient population. And these advances coupled with the availability of large amount of data with the machine learning advances, now what's possible is actually to take large amount of really heterogeneous data. I'm going to give you some examples. Apply machine learning methods that instead of thinking of heterogeneity between data sets as a curse, it considers it a blessing in disguise, and finds diagnostics that are really generalizable despite patient's age, host genetic background, the environmental exposure, sex, the strain of the pathogen, irrespective of across different studies at what time point the patient samples were collected, the comorbidities they may have. It accounts for these methods now really allow us to account for this heterogeneity upfront when we are doing a biomarker discovery, instead of making it an afterthought of about a diagnostic biomarker being locked and when we are trying to validate in independent cohorts. So how do these methods usually work? And here is the first application in the field of sepsis, where we started with 24 cohorts that came from really large number of patient population. This was a fantastic collaboration actually with Hector. And we were able to show that despite all of the heterogeneity, there were an 11 gene signature that distinguished inflammation due to infection from a sterile infection. And we were then able to follow up and show that even when you go in even a larger cohort of larger number of studies that are even more heterogeneous, these transcriptome-based signature or a host response-based signature continues to perform just as well. There is no overfitting in a statistical speak, or another way to say it, it continues to perform with the same accuracy in completely unseen, prospectively collected cohorts. But then you can say, well, this is us hyping up our own work, right? So there have been prospective studies by independent groups showing that these host response signature continues to perform with the model that was described in a previous paper, that they continue to perform with the same accuracy as they were described in the original paper. And then the paper on the right in a critical care medicine literally came out yesterday after I submitted my slide. So again, the prospective validations of these host response-based signatures are starting to continuously show that immune system can really do what it's supposed to do. But we can even go further. We can now start adding machine learning models that really consider nonlinear relationships between these mRNAs. And you can apply these models, locked models, with preset thresholds, just as we would want to do in a clinical practice, and then apply and then validate in prospectively collected cohorts on a completely different platform to show that they still continue to perform very well with very high specificities and sensitivities. But in sepsis, just as Philipp was talking about earlier, the antimicrobial resistance, simply knowing that somebody has infection is not good enough. We also need to know whether they should be treated with antibiotic or not. So there have been a large number of transcription signatures described for identifying whether a patient has a bacterial infection or a viral infection. And when you look at these signatures and how they were discovered, they again used just by the virtue of the kind of the groups that were really working on this, turns out that each one of these signatures are usually defined using patient populations in the Western hemisphere. So what happens is it's more extracellular bacterial infections that are prevalent, not the intracellular bacterial infection. So when you use these same signatures and apply them to patients with intracellular bacterial infections, their accuracy dramatically drops to a point where a lot of intracellular bacterial infections may get diagnosed as viral infection, and we cannot really reduce the antimicrobial resistance. So, but given that now there is a large amount of data available, and there is participation also increasing from patients from the other countries, we can now, and we have been able to now take even larger number of samples than before, thousands of samples simultaneously, tens of cohorts across multiple different countries, and identify host response that is even more generalizable, that truly leverages what the immune system has learned over millions of years to say, this is a bacteria, this is a viral infection, this is a conserved host response that I need to use and respond to this pathogen, and really show in a completely prospective cohorts from different countries than the ones that were used for identifying these biomarkers, and show that they are still continuing to identify whether patient has a bacterial or a viral infection with sensitivities and specificities in a 90% range. But then, just as Tom was saying, this is the diagnosis. The next step is to figure out whether a patient, what is going to happen to the patient at the time of presentation. So this is another paper that just came out in Immunity earlier this month, actually, last week, where we were able to take thousands of viral infection cross-sectional studies and use that to show that at the time of presentations, we can identify what trajectory the patients are going to be on, which allows us to predict at the presentation whether a person with a viral infection will have a severe infection or a mild outcome. And depending on that, we better triage them. But what is even more important is when you look at these patient trajectories, you start to realize that there are two types of broad categories of host response. There is a detrimental host response and a protective host response. So now you can start imagining therapies where on one side you have a drug that suppresses detrimental host response, and on the other side, it enhances the protective host response, which we can imagine in the long run would start improving the patient's outcome overall. So to summarize what I just told you, really, host response can allow us to detect presence, type, and severity of infection. These host response signatures are now starting to be implemented onto a point of care devices. For example, and I'm a co-founder of Informatics, so that's my conflict of interest. But we have now developed a device with a sample to answer cartridge that with a 30-minute turnaround time that can measure up to 64 genes simultaneously. So you can imagine a single cartridge that is able to measure each of these diagnostic tests in a single cartridge with 30-minute turnaround time and be able to respond to more than 2 billion cases of infections worldwide every year. And that's the potential that these mRNA signatures are starting to provide. I'll stop here by thanking the people who really do this fantastic work. Allow me to take the credit. All the collaborators who really support our computational analysis by enrolling patients, doing the trial, and showing that we are really starting to move diagnostic into this new phase. And the funders, thank you.
Thank you very much, Purvesh. That's really fascinating and very coherent overview of the topic. I think it's really exciting that you're able to leverage these really large and different data sets. And that brings up the question that has come up in the chat here. How do you discriminate between biological heterogeneity and technological heterogeneity? I guess that means in terms of exactly which tests are done, or perhaps the technologies involved, if you're combining these multiple cohorts.
Right. So that's a great question. I always get asked this question that there is so much heterogeneity that how do you know that your results are not confounded by certain things? And the answer, and it's not a coward's answer, but it is me trying to challenge a different way of thinking. While we've been told that the heterogeneity is a curse, it is not a curse. It's actually a blessing in disguise that if a biological signal is true, it should exist irrespective of whether a patient is a 2-year-old or an 8-year-old, whether it was measured using an RNA-seq, a microarray, or a PCR. If the signal changes depending on what technology you reuse, it may not be really a robust technology that we can apply to a global problem such as antimicrobial resistance. So our approach is not to correct for any of those, but actually account for those heterogeneity and say that I am looking for a signal when we are doing our discovery analysis, and look for a signal or look for a set of variables that are consistently able to discriminate between the two groups that we are interested in, irrespective of whether the patient was young or old, man or a woman, had a comorbidity or not, whether they were measured using one technology versus the other. And that is what the advances in machine learning have really allowed us to do, to instead of worrying about reducing the heterogeneity, embrace it and leverage it to find more robust diagnostic signals.
Okay, great. And just very quickly, and this is all about finding global solutions for a global problem. Are you aware of any challenges getting a really heterogeneous data from different settings? I'm guessing most of the data sets you work with come from high income settings at the moment.
That's a great question. So I would have 5 years ago, I would have said, yes, that was a problem, but not anymore. There has been more awareness that we really need to bring in more heterogeneity. And for those of you who are interested in doing anything like this, the NCBI has a gene expression omnibus database where it now has more than 3 million transcriptome profiles publicly available for anybody in the world to use without signing any IRB. Everything is anonymized. And we've been able to show across a broad spectrum of diseases, and that includes organ transplant, autoimmune diseases, vaccinology, that there is sufficient data available. And once you have 4 to 5 data sets for a given disease included, usually your signals tend to be really robust, and you've typically accounted for most of the heterogeneity.
Thank you very much for that talk. So we'll now move on to our next talk of this session. I think really it's almost been introduced by Professor van der Pol earlier. Our next speaker is Hector Wong. And as you know, and you probably heard, he has already been introduced regarding the topic of predictive enrichment and his work in that area. So Hector is a professor of pediatrics at Cincinnati Children's Hospital Medical Center and the University of Cincinnati College of Medicine. He is also the director of critical care medicine at Cincinnati Children's Hospital Medical Center and Cincinnati Children's Hospital's Research Foundation. And his research is focused on translational research in sepsis, but particularly utilizing a large multi-institutional genomic and clinical database of pediatric septic shock. And Hector's going to talk to us particularly about this idea of Theragnostics and phenotyping for sepsis trials. So it gives me great pleasure to introduce Hector.
Thank you, Louise. I'm going to assume that you can see my slides and hear me unless I hear otherwise from you. So good afternoon, good morning, good evening to everyone. It's good to be here with such a large audience committed to combating sepsis. So hopefully this is of interest to you and it'll build on some of the previous talks. So these are my disclosures: funding from the NIH. My institution and I hold a number of patents related to sepsis biomarkers, and I'm on the scientific advisory board of these two biotechnology companies that are dedicated to sepsis. So let's get back to this important concept of enrichment that Dr. van der Pol introduced. Again, by way of reminder, enrichment in general refers to the use of any patient characteristic to select a population in which your intervention is probably more likely to be effective when compared to an unselected population. And then there are two more specific forms of enrichment. There's prognostic enrichment, which is the selection of a patient population more likely to have a disease-related event such as mortality. And to follow up on what Dr. van der Pol said earlier, I think it is accurate to say that previous trials have used a form of prognostic enrichment based on clinical criteria, but that we can do better, and that perhaps biomarkers can enhance our ability to conduct prognostic enrichment. And then also, as Dr. van der Pol indicated, there's the concept of predictive enrichment, which means that you select a patient population in which your intervention of interest is more likely to be effective based on underlying biology or physiological mechanism. And I couldn't agree more with Dr. van der Pol, at least to what he was alluding to, is that to me, these are the key concepts or the key enablers of realizing the promise of precision medicine for sepsis and other forms of critical illness. So this is another cartoon version of what Dr. Vanderpoel showed earlier. Imagine a heterogeneous sepsis cohort. Each dot there is supposed to be a patient, and each color is a subset of those patients. And that perhaps we should be doing something analogous to this, in that we first divide the patients using prognostic enrichment. And I would argue that those who are low-risk patients probably are not going to benefit or derive much of a benefit from an experimental intervention. And so those patients perhaps should be parsed out and treated with standard of care and not be enrolled in some of these higher end trials. And then that leaves you with the higher risk patients, and then perhaps those patients could undergo a form of predictive enrichment, which selects them for treatment with therapies that are targeting a specific biological mechanism, but it's more likely to be effective because you've presumably selected those patients based on commonality of having that mechanism that can be targeted. And so I'll show you a few examples of that. One is this thing that we call Persevere, which is something we call the pediatric sepsis biomarker risk model. And it's a serum protein biomarker-based decision tree. And the way that we got to those biomarkers is some of the transcriptomic work that Dr. Khatri referred to. The tree is really not that complicated. It's based on classification and regression tree methodology. And what it allows us to do is to provide a baseline risk of mortality among children with septic shock. So this is a form of prognostic enrichment. And to again, to build on what Dr. Khatri said, this is at the time of presentation. This is when we measure those biomarkers. So to emphasize what he said earlier, I think that's the time where I think it's probably most critical to be able to predict trajectory or estimate the risk of a certain trajectory. This is what the tree looks like. And so based on biomarker cut points and a series of binary recursive partitioning decisions, patients end up classified into these terminal nodes. And so patients that end up in one of those four terminal nodes have a very low risk of mortality, ranging from 0, which is probably not true, to 0.019. And then patients who are allocated to these two terminal nodes have an intermediate risk of mortality as listed there. And then finally, patients that are allocated to those three terminal nodes have a pretty high risk of mortality, ranging from 0.297 to 0.44. Keeping in mind that overall mortality for pediatric septic shock is about 10%. And so we use those classifications to estimate the risk of mortality. And this is the most recent paper which applied these decision rules a priori. To use a term that Dr. Khatri used, we locked the decision rules and applied them to a prospective cohort, and these were the performance characteristics. Also, to build on what Dr. Khatri said, this was a very, very heterogeneous group, or a very confounded group, if you will, because it ranged all the way from 1-week-old babies up to 18 years of age. It included kids with no comorbidities, kids with cancer, kids with transplantation. So very, very mixed bag of patients. And so similar to Dr. Khatri said, we embraced that heterogeneity or that complexity to see if this model could perform well, and we were happy to see that it did, even under those difficult conditions. This is just a representation of the same data. It broadly groups the patients into low, intermediate, and high risk as predicted by this model, and you can see that the survival curves are quite different across those three groups for 28-day mortality. If you're interested in these data, they're published in this paper that appeared at the end of 2019. This is an analogous approach. This was a really neat approach that included 3 different investigative groups, including Dr. Khatri's group, where they took essentially all of the available transcriptomic data that was out there publicly and used different approaches of machine learning to come up with mRNA-based prediction tools for mortality. And one of those now, as Dr. Khatri alluded to, is almost ready for prime time by way of rapid diagnostics. This is another example. This is again Dr. Khatri's group and Tim Sweeney's group that they've applied this this classifier, this mRNA classifier, to patients with viral infections secondary to COVID-19, and they were able to predict pretty accurately their trajectory and risk of poor outcome. So those are examples of prognostic enrichment. There are many others. Now we're going to turn to predictive enrichment, and this is, Dr. Vanderpoel alluded to this. And here what I'm going to emphasize is what I call gene expression-based endotypes of septic shock. I would submit to the group that sort of single biomarker attempts at predictive enrichment are clinically attractive, but I would say that they're probably biological wishful thinking. I think septic shock is much, much too complex to be able to perform predictive enrichment based on a single biomarker, but perhaps that's something that we can discuss. As I said, I understand the clinical attractiveness of that, but unfortunately, I think life is more complicated than that. And so these are examples of 4 different papers. Starting chronologically in the top left is Julian Knight's group, which described a transcriptomic signature that could group patients into two gene expression-based endotypes. The next paper is from Dr. Vanderpoel's group, which used a similar approach, and they described 4 groups. The next paper on the bottom left in 2018 is from Dr. Khatri and Tim Sweeney, in which they used all publicly available transcriptomic data to generate 3 classifiers for endotypes based on gene expression. And then on the bottom right is our work back in 2015 describing 2 endotypes of pediatric septic shock. And so there's important commonalities across these 4 strategies. So they were all used or developed using whole genome transcriptomic data, sort of an unbiased discovery approach. They all showed association between subgroup membership and outcome, and they all describe a subgroup that has gene expression patterns corresponding to the adaptive immune system. So it's interesting that 4 different groups have come up with this similar classification, albeit by different genes. And so I think this is really important because, as was alluded to earlier, there's a lot of talk now about using immune-enhancing therapies for sepsis. And so if one believes that these endotypes are real, for lack of a better term, one could envision that these types of therapies are going to behave very, very differently depending on which endotype one belongs to. And so one of the simple questions that we've asked is, could these endotypes account for treatment heterogeneity that we see with adjunctive corticosteroids? So, right, there's been trial after trial with septic shock and adjunctive corticosteroids, and we still don't really have an exact and clear answer as to benefit, and that probably has to do with treatment heterogeneity, and that perhaps these endotypes might account for some of that. So this is the VANISH trial. This was an adult with septic shock that there was a 2x2 design randomizing to vasopressin and norepinephrine. And then once patients landed on a certain dose of vasopressor, they were randomized to hydrocortisone versus placebo. A subset of that group had transcriptomic data, and then the investigators classified those patients using the transcriptomic data to SRS1 or 2, and they tested for an interaction between corticosteroid exposure and SRS assignment. And what they found here is that among patients who are SRS2, corticosteroid exposure was associated with an increased risk of death. And again, this is in the context of a randomized trial. This is analogous to our data that we published several years ago, in which we showed that in kids that are allocated to endotypes A or B, corticosteroids are actually independently associated with increased mortality among endotype A patients. And when I say independently, I mean that we were able to at least account for illness severity and those kinds of things, age and comorbidity. So the signal seems to be strong, and we've been able to replicate that signal in various subsequent studies. This is a marriage of those 2 studies, right? So as I said, the VANISH transcriptomic data is publicly available. We downloaded that data, actually with Dr. Khatri's help, and we assigned patients to pediatric endotype A or B. This is what the vanished patients look like. At the top are what we call our reference mosaics for endotype A and B. And in the middle and last row are examples of vanished cohort patients who were classified into either endotype A or B. And so we use this classification system with the vanished cohort, and we looked for an interaction between endotype assignment and hydrocortisone. Here, the reference group is endotype B and placebo, and what we're showing here in the bottom is that among endotype A patients who were randomized to hydrocortisone, the odds ratio for mortality was higher, just on the edge of significance there, reflecting a relatively small population. But the important take-home point is that we were able to use this endotyping strategy in an external cohort of adults, and the data, or at least the trend that we see with hydrocortisone, held up. So we were very pleased to see that. And so it's one thing to say who shouldn't get corticosteroids. It's another to say, well, who might benefit? And so what we've done is that we have combined prognostic enrichment with predictive enrichment with the technologies or systems I showed you earlier. And what we found is that endotype B patients, and these are children who are at intermediate to high baseline risk of mortality, when those patients are prescribed corticosteroids, there's a more than 10-fold decreased risk for poor outcome. The caveat to these data is that they're all observational, right? So the corticosteroid prescription was at the discretion of the team caring for the patient, so the steroids were not randomized. And so there is lots of potential there for confounders. And so we're testing this prospectively now. This is the Schütz trial, which is a randomized placebo-controlled trial of hydrocortisone among kids with septic shock. We will be collecting biological samples for prognostic and predictive enrichment. We were not at the stage where enrichment can actually inform enrollment, but we do have a pre-specified post-hoc analysis where we will analyze the outcome data based on stratification and enrichment. Dr. Ronat's group in France is doing a very similar thing, the Records trial, that's trying to do this same kind of predictive and prognostic enrichment in adults with septic shock who are randomized to corticosteroids or placebo. So more to come on that. I hope that that was of interest and that dovetailed well with the other speakers, and happy to answer other questions. So thank you for your attention.
Thank you very much for that talk. And yes, I would say it dovetailed perfectly with the speakers before. So thank you, Hector. I think this area is really exciting and moving very fast. It's great to see that there are now some prospective trials underway. I guess my question is a bit broader then, assuming that the prospective trials confirm the positive effects, signs we've seen in the observational studies, do you think there will be any barriers to people adopting these new tools? Is there likely to be any hesitancy, do you think?
There's always hesitancy to new things, right? So there's a whole science around that, around implementation science, right? The barrier that I've been trying to deal with is more the technological side, right? So these, especially the RNA signatures, in order for them to be effective, we can't take 24 hours to get the data back, in my opinion. We need these data back within an hour in order to inform a clinical trial and clinical decision making. I'm at the bedside like many of you, so I get the practicality of actually doing this at the bedside. And so I recognize that rapid diagnostics is a key to this. So to me, that's the biggest barrier. And as Purvesh alluded to earlier, I think his group is very well on their way to having a rapid platform that's technically friendly, accurate, reliable, and allow us to do this kind of endotyping in the ICU setting where we need that to be done quickly. And up to, I think Purvesh mentioned 64 genes, so that's impressive. So I'm really looking forward to more of their work and hoping to be able to piggyback onto some of that work.
Yeah, I really agree. I mean, it's amazing really to hear that there seems to be something that's actually potentially going to be clinically useful in real time, available very soon, at least in a clinical trial context. So thank you very much for that talk, Hector. And I guess with that, we'll move on to our next talk, which really is about a project, a very practical project, which is aimed at filling the gap until these new technologies are available rapidly and at a point of care. And our speaker is Jean-Baptiste Ronat. He graduated in biology and biotechnology laboratory analysis at Lyon University. And he's worked in auditing and implementing laboratory capacity strengthening projects in various fields of biological diagnosis in low and middle income countries for Médecins Sans Frontières and the World Health Organization. And he's got a particular interest in clinical bacteriology. So as I said, he's going to talk particularly about what he describes really as a bridging project, the revival of robust manual solutions to sepsis, diagnostic and AMR surveillance in low resource settings. And this is the mini lab project by Médecins Sans Frontières. So very welcome, Jean-Baptiste, and the floor is yours.
Thank you so much. And you can say Doctor Without Borders as well for those that are not French speakers. Good afternoon, good morning, ladies and gentlemen, the organizer. I'm going to present what we are doing at Médecins Sans Frontières to adapt and revive common bacteriology technology to provide, as it was mentioned, to our practitioner access to clinical bacteriology laboratory to better diagnose sepsis and provide AMR surveillance in low resource setting while we are waiting for such a wonderful technology that were described before. So I will firstly very briefly discuss about the re-emerging threat of sepsis and AMR in low resource setting. Then I will present you briefly the mini-lab concepts. And then I will focus on the validation study we have made on the blood culture technology we selected. So as you are very aware, and I will not dig into detail, the sepsis is a preventable disease that life-threatening condition marked by severe organ. And in 2017, it was estimated that it had affected 49 million individuals and was related to approximately 11 million potentially avoidable deaths. Sepsis mortality is often related to suboptimal quality of care and inadequate health infrastructure, poor infection prevention measure in place. And antimicrobial resistance further complicates sepsis management across all settings, particularly in high-risk populations such as neonates and patients in intensive care units. In a recent review, it has shown that among bloodstream infection, non-typhoidal salmonella was a major cause of global mortality, especially in low-resource setting, with highest incidence in Africa. Another recent study showed that Sub-Saharan Africa and Southeast Asia are the most affected region by sepsis, and that the highest mortality is in neonatal and pediatric population. And this geographical area and population are where MSF is intervening more often. While etiology data for neonatal infection are very scarce in Sub-Saharan Africa, especially in bloodstream-related infection, what the study really describes is the rising concern of a bacterial being resistant to WHO recommended antibiotic treatment. Unfortunately, today, the only way to diagnose sepsis or other bacterial infection and survey resistance trends in population is by implementing clinical bacteriology laboratory. And it's massively recommended by WHO to implement such laboratory, starting from district hospital laboratory, while point of care that were described before should be implemented at primary care setting. However, the issue, we faced many challenges in implementing those type of labs in low resource setting. They require stable electricity supply and control environments. Logistics is really a challenging and a nightmare. They are composed of many reagents requiring cold chain with short shelf life. Also, conventional methods such as automatic blood culture machinery, they have not been adapted for those environments, and too few rapid diagnostics tests for bacterial detection are available. Then one of the major issues as well is then finding skilled microbiologists or experienced laboratory technicians require to work in those laboratories is very difficult. And these are just few examples. MSF has a standardized approach to surveillance and data in its own standalone laboratory or when working in partnership with another lab. However, some sites have so little access to microbiology that improving it's a key component of AMR response in itself. So leapfrogging solutions are much needed in this context, such as rapid, affordable, and effective point-of-care diagnostics, like it was described before, that can identify pathogen and provide antibiotic susceptibility testing. Yet, these breakthrough, affordable, low-maintenance, robust products have not yet materialized, mostly because of market failure, but as well barrier to use. As a result, MSF decided, while waiting for this technology, to develop its own leapfrog solution, and the Minilab. We have adapted manual bacteriology technology, focusing on the ease of use, robustness, and clinical relevance in resource-poor contexts. Its goal is to be a turnkey solution that is self-contained, easily installed, quality assured, and adapted to low resource setting, and can be operated by trained laboratory technicians without prior microbiology expertise. The first Minilab prototype focused on diagnosis and ST for bacterial bloodstream infection, and it can be rapidly deployed and set within 1 day. It is composed of six foldable sturdy bench. That is a sturdy transport box that transform into a standalone laboratory bench with all the material packed inside. The modules are plug and play with washable surface. They integrate electricity protection and can be deployed in less than 20 square meter room or tent or container and provide all safety requirements. To adapt clinical bacteriology to lab technician, to non-experts in microbiology, we had to tackle several aspects. As mentioned just before, we had to develop box bench platform to protect and transport selected equipment for rapid setup. Then we have to adapt, simplify analytical process from test device to equipment used and safety measures. And then guide user along the process with clear pictogram, graphical layouts, signposting. And finally, we had to adapt didactic document and training, such as SOP, manual for the trainers, e-learnings to have rapid access to all the information. And then we have created for the result interpretation and control, a laboratory information system for the workflow management and microbiology decision support system that integrate all the rules that microbiologist knows by heart into a system that will interpret one user in case of error. These different components have been individually evaluated extensively in different studies that are under publication. And I'm just going to present you today what we've did on the blood culture system, but very briefly, what we did to adapt the workflow is that we selected a biphasic blood culture bottles, and I'm just going to describe after, but for example, with Beckman culture, we developed a unique microplate to have a phenotypic identification of the bacteria instead of having 2-3 different gallery. We had as well developed 3 microplates based on the microscan technology for AST testing using MDR indicator of the glass from the WHO surveillance system. And we had as well developed a probabilistic-based algorithm for pre-identification orientation and adapted several other technologies that I won't be able to develop and present you today. So while the Minilab has been deployed in IT and is now under evaluation on the second field pilot in an MSF-supported hospital in Central African Republic, I'm going to present you the study we have made to validate the blood culture bottles. So you are aware that manual blood culture bottles are frequently used in low resource setting where automated alternatives are less suitable. The manual type of BCB that we selected was done after definition of specification and massive comprehensive market review, and we selected biphasic methods, so which is used to be named Castaneda bottles, which is an old technology, because in general they have better recovery for gram-positive species and faster bacterial growth compared to monophasic. Yet the issue is that performance evaluation of this BCB are lacking, and especially in comparison to automated system. So what we did, so we selected 3 biphasic BCB on the markets, and one where they were evaluated in a proof of concept prior to this study, and the final choice was the bi-state biphasic bottle from Autobio Diagnostic from China. And as well, we kept the use of BacT/Alert read manually, because actually it's widely done in Africa, where they use the BacTech or BacT/Alert or other automatic blood culture to read it visually. We use the BacT/Alert and TOMATE as a reference methods, and we report here some of the findings of this evaluation. So what we did, we spiked in triplicate with more than 177 clinical isolate coming from low resource setting, representing pathogen frequently found in low resource setting for both adult and pediatric population. And the growth in manual BCB was evaluated by visually inspecting the broth agar land twice daily. And for BacT/Alert blood culture, the color change of the growth indicator was selected. So what we've seen in our study is that the Autobio bottles perform very, very interestingly in terms of yield of positivity. So we had 95.9% positivity rate as compared to the automatic bottle, which was around 96%. And in terms of speed of growth, we had a better speed of growth from the Autobio as compared to the manual read BacT/Alerts at D1 and at D2 as well. There were no systematic failure on the different bacteria that we seed into the blood culture bottles. So in this in vitro experiment, the yield and time to detection of manual BCB were comparable to an automated system. Bi-state BCB outperformed the manual visual read BacT/Alert bottles, and visual reading of the bi-state BCB was facilitated by the use of a BCB light box. So we developed a light box with LED technology to provide a more illumination of the broth. And recent evaluation done, which is published on the field in MSF Hospital in IT on 450 samples, confirmed the ease of use of the Autobio bi-state BCBs. So in conclusion, I would like to say that clinical bacteriology has really a high added value in low resource setting for sepsis diagnostics. Manual blood culture are widely used, but not many studies on performance are available. And in low resource setting, in absence of 24-hour laboratory service, manual blood culture still has a high added value to tackle sepsis, taking in consideration that they are cheaper, robust, have longer shelf life. They are around 18 months to 24 months as compared to 12 months, and require less maintenance as compared to automatic system. And when deployed, the Minilab will be able to generate standardized, representative, high-quality data on pathogen and drug susceptibility from the target population, and we hope to improve patient clinical management. And I'd like to thank you.
Thank you very much, Jean-Baptiste. That's a really excellent talk, and really interesting to see something so practical happening. And as I said at the beginning of this session, the overwhelming request from certainly yesterday's speakers in the majority of sessions was that we improve access to diagnostics throughout the world if we want to improve sepsis outcomes. And this project looks like it's really attempting to address this issue.
Thank you.
I have a question really, is you're beginning to pilot this system now. How have you selected the site where you're going to do that, and what are your plans, assuming this is successful, for rolling out and scaling up?
Thanks for the question. Actually, we selected the sites based on the population that attend the hospital, and based on the strategy that MSF defined to provide access to clinical microbiology, because we intend to provide access in hospital where there are population at risk, like children, neonate, malnourished children, or patients living with AIDS and other population at risk. So the first site was selected in Haiti because it was as well a burn center, so and burn tend to be more at risk of developing sepsis. And the second site that we selected, which is an hospital, a pediatric hospital in Central Africa, it was selected as well because it's part of the population at risk that are identified in MSF as really a priority.
Okay, thanks. We just got one question quickly in the chat saying, have these blood culture bottles been evaluated with respect to neonates?
We have done on pediatric formulation, but not on neonate, not in the studies that we have done.
Okay, thank you very much. Thank you very much, Jean-Baptiste. The next talk sticks with our theme of diagnostics and technology, but actually is transiting more over into the therapeutics area, which is the topic of the next session. So I'd like to introduce our final speaker, and that's Professor Alison Holmes. She is a professor of infectious diseases and director both of the National Institute for Health Research, Health Protection Research Unit in Healthcare Associated Infections and AMR, as well as the Centre for Antimicrobial Optimization at Imperial College London. She leads a very large interdisciplinary infectious disease research program focusing on antimicrobial resistance, epidemiology, public health, precision medicine, development of emerging innovative technologies to address infection prevention and management, antibiotic optimization, and antimicrobial resistance. And that's both nationally in the UK and globally. So welcome, Alison. And Alison's going to be talking about harnessing digital technologies for sepsis diagnosis and decision support.
Great. Thank you very much indeed, Louise, and hello to colleagues around the world. So I'm going to be talking mostly around the perspective of how we can optimize antimicrobial therapy through the use of innovation, technology, and of course, harnessing the potential of artificial intelligence, so that we can really improve how we can use our existing agents more effectively, and also tackle our very tricky resistant infections, and enhance and preserve our existing agents, but also how we can be much more responsive and dynamic in how we use our antibiotics. So just thinking about our own history of developing decision support. So we've moved from developing paper guides that could fit in people's pockets, to developing smartphone apps, to also considering the kind of social science around developing some of these decision supports. So what they call a kind of boundary object, which enables different social groups to engage around something that means different things to different groups, and how we can actually use the decision support to engage multi-professional groups in antimicrobial prescribing. And then also, how can we use incentives in terms of gaming to raise awareness and improve practice, and also use the data from such initiatives to improve practice. So moving on from having a rather static project around looking at optimizing antimicrobial choice, we looked at, of course, how we can integrate multiple data sets to improve our antibiotic decision making, and also making it user friendly. And indeed, you know, now the kind of tablet is, of course, part of the team, and we really need to think about how we engage with colleagues far more effectively so that they find these tools useful and they fit in with how people work and make decisions. And a lot of work was done on looking at what people needed and existing antimicrobial decision support systems. And this is a lot of work done by Tim Rawson here on looking at what were the challenges in decision support and how could we do a lot better. So there's marked lack of flexibility in terms of decision support systems, in terms of recognizing inter-individual variation. There's major gaps in considering dose optimization and extremely little engagement in end users and almost negligible patient and public engagement. They're very static and they don't actually think about the data related to how people make decisions or how they like risk projected to them, and very little co-design, which we can do much better at dealing with. And also future-proofing in terms of integrating with any emerging diagnostic platforms, and the consideration that actually, even although we think infectious disease is the most important thing, we really do need to recognize that comorbidities do exist and polypharmacy really does exist. So we tried to think about how best to integrate all of this into a kind of new concept of antimicrobial decision support that pulls much more in and also considers dosage much more effectively and engages with the populations that need to know about this and shape it. So that led to a variety of pieces of work and outcome which tested supervised machine learning for infection risk and also developed decision support systems that were highly effective at identifying bacterial infection in cohorts of patients, but additionally reflected upon the kind of nature of the data required, the ethics of it, and also the transparency. And I think this is something we need to dwell on quite a little bit more. We're delighted that the decision support that was developed was CE approved, which was great to see. But then thinking about COVID and does this matter in COVID? I mean, does it work in COVID in terms of identifying co-infection? So our existing decision support system was not quite as good pre-COVID, but with just some addition of existing routine data, we were able to successfully identify co-infection in the context of COVID quite successfully. And this article was recently published in March. And just to flag, this is routine data. This is nothing fancy. And also it does not contain procalcitonin, but it was routinely collected data that was used for this. So moving on to dosing, which is something, although I see, there's been a lot of talk about artificial intelligence and different applications, I don't think there has been quite so much in the kind of considering of how we should be dosing our antimicrobials. And, we know that there's massive heterogeneity, there's huge inter-individual variability, but particularly in sepsis, there's massive intra-individual variability that is really not adequately addressed in terms of how we dose our antimicrobials and how we dose them in increasingly tricky and resistant infections. So we then need to think about how we can look at dose monitoring. And most of us think that therapeutic dose monitoring is extremely boring and really, really challenging and very difficult to do. And we're, you know, it's a logistic nightmare. So within the box, you can see the kind of pathway from taking blood from a patient, getting it to a lab, checking it in, getting it analyzed, reported, reviewed, interpreted, and then fed back to the prescriber, which goodness knows how long that might take. It might be far too late to be able to influence any sensible decision making. And furthermore, that the dose adjustment that will be made may be based on large level population estimates, and you're using that to apply to individual. So around the edge of that box in purple, you can see that some of the hurdles are identified, such as I said, population level estimates are being used. Timing of samples is tricky, stability is tricky, and I mentioned the delays in reporting, and there may not actually be a valid assay anyway for that agent. So that's why we all think this is far too difficult. But let's think about what we could do. And so here's a little kind of wish list of how we can improve things. So we want to get to the stage where we can have data to optimize antimicrobial use and tailor the dosing for the individual, the organism, the infection, and also the resistance mechanism as well. We want to be able to do this dynamically in real time, and we want this non-invasively. So there are technologies that exist. So let's have a little think about the real-time drug monitoring. And we know that this technology already exists in terms of what can be done in terms of closed-loop control. And indeed, we're delighted to be working with some of the pioneers in glucose monitoring and the artificial pancreas. And indeed, that's where much of this technology has arisen from. And we're shifting that to the application in optimizing antimicrobial dosing. And it's also being used in anesthesia as well. But so that's the kind of real-time, that's the kind of closed-loop control that can exist around antimicrobial dosing. But what about improved technologies for drug monitoring? So I said we need to do something minimally invasive. Taking multiple blood samples or leaving indwelling intravascular devices is not adequate. We want things at the point of care, but that also can provide continuous monitoring and also look at a broad range of agents. And one of the technologies that is looking to have major potential is the use of microneedles and microneedle electrochemistry sensing technology, which is providing a means of doing something at the point of care and that can provide real-time continuous monitoring, no blood sampling required, opportunities, of course, in pediatrics as well, and minimally invasive and looking at the dermal interstitial fluid. So you can see in the picture that the microneedle patch is about the size of a postage stamp and just sits above the skin. So how can these be used? Well, they already have been, and we've managed to turn microneedles into biosensors, initially for the monitoring of penicillin. And this was by using the enzyme beta-lactamase as the means of being able to sense a beta-lactam by the action of the beta-lactamase hydrolyzing a beta-lactam to acid and a proton, and the microneedle surface being used as a pH sensor, and it can detect decreases in the pH caused by the hydrolysis of penicillin at the sensor surface, and the changes in pH are detected as voltage changes. And so this was developed, and proof of concept was published, but I'm pleased to say that we rapidly could move from a proof of concept to evaluation in healthy volunteers, and this was published by the Lancet Digital Health at the end of 2019. So this was really exciting that this actually works, and it's technology that is easy and it can be produced at scale and is can be relatively cheap. So that's around the kind of dose monitoring and the opportunities of these microneedles. I've talked already about closed loop control, and this has also been developed and investigated and tested for beta-lactams. And as well as the opportunities for closed loop control, which is just for the individual patient. Of course, anytime these microneedles that are connected wirelessly are used, they generate data to inform dosing of a variety of specific agents and for different clinical scenarios and conditions and organisms. So this is really exciting, and I think there's going to be major progress made in this area over the next year or two. I'm not just looking at enzymatic biosensors, but looking at aptamers, and these are like kind of monoclonal antibodies, but DNA and RNA sequences, which are now available for a broad range of agents. And we are certainly now working on a range of aptamers related that we're using for looking at gram-negative agents and also for antifungals. So I think this is going to be a really exciting area. It's also interesting in terms of this technology to combine different agents on the same postage stamp microneedle platform. And we're also exploring how this can be combined with biosensing of host factors. So there's a range of other ongoing work that I don't have time to go into much at the moment, but integrating the dose optimization also with rapid diagnostics and harnessing much more data from diagnostics by the application of artificial intelligence and molecular biology. For instance, multidimensional analysis of existing PCR curves are already helping us with identifying a range of different carbapenemase producing organisms. And this is work by my colleague, Jesus Rodriguez Manzano up there, and also integrating it with physiological monitoring. And there's been much more talk about this as well in this Congress, but there's work existing with colleagues in Vietnam looking at continuous non-invasive monitoring in dengue, which of course is completely applicable to our the context within sepsis. And I think bringing all of this together is going to be incredibly exciting. So thank you very much. And I've got masses of people that I work with who bring a range of skills together. Thank you. Thank you, Louise.
Thank you very much, Alison. That was a really comprehensive and kind of whirlwind tour through many areas we haven't even touched on yet in this session. I think one of the comments here, well, many comments saying excellent presentation, and I would very much agree with them. I think you've touched on things which are really important, like how to engage end users with co-design. You've talked about approvals for clinical decision support tools. And of course, then you brought in this whole area of precision and personalized therapeutics. I guess we're nearing the end of the session. And I guess my question probably to you is if you have to pick one area which you think is going to see the most rapid development towards real clinical use and utility, what would your guess be?
Oh, okay. I'll have to select one. Well, I think we're already there with the decision support, Louise. I think whilst everybody's focusing on data, we're not actually focusing on the people and the kind of qualitative data about how people use decision support. But I think that's already there. But I think in terms of the technology, I think the opportunity for the use of biosensors for both drug monitoring and host response monitoring is going to be massively advanced over the next 2 to 3 years.
Great, thank you. And so I think at this point, I will draw this session to a close. I would like to thank all our speakers. They've given some excellent presentations. They've stimulated a lot of interesting chat and discussion in the dialogue. And we're very grateful for your time preparing and presenting to us today. I'd like to thank all the participants. It's amazing looking at the chat to see how many countries, people are in so many countries and engaging in this session as well. So thank you very much. As I said earlier briefly, everything has been recorded, and over the coming weeks, these sessions are going to be released either on to YouTube and also Apple Podcasts, so keep looking for those. Please continue to listen to the final few sessions of the Sepsis Congress. They're moving on to therapeutics and management now. And of course, interact via the World Sepsis Congress and Global Sepsis Alliance websites. So again, I thank sponsors, I thank the organizing committee, and I thank our speakers and of course the audience. Thank you.